All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So we'll continue our discussion. I wanted to today talk about Maraviroc. However, this news about the efficacy of vaccines and convalescent plasma and Regeneron against the Indian variant or 617 and 618 variants that originated in India is a very important news as well to go over. So I thought I'll do that. So in the discussion today, number one, we'll see a number of things. Number one, what is the increase in infectivity by the Indian variant, so 617 and 618? What is the shedding increase by these variants? Number three, what is the efficacy reduction by the convalescent plasma, by vaccine-generated antibodies, and by Regeneron antibodies? And finally, what are the conclusion of the authors? Are they concerned about it or not? This study is done in New York University, so it is pretty recent and pretty important study. Generally, if I can give you one comment here, I would say that, yeah, there is no worries. Actually, other variants have been even stronger in their reduction in efficacy or in their spread than the 617 and 618. So not much to worry for these variants. What would happen in the future variants, we do not know, but for these ones. So let's start our discussion. Margaret, welcome. And we will then go from there. So as I was on a few days of vacations, I did some paintings as well. So this is a watercolor painting that I did. It's a digital watercolor. It is done in Adobe Fresco. I just did it very quickly. It's more of a sketch-like thing than the watercolor. And total credit, one of the figures here are actually from another artist's sketch, and I from their teaching book. So I think they meant it to be drawn, but I thought both of them together make a great painting. The title of this painting is That You? So that is the title. So this is the latest painting. Now here is, for your viewing pleasure, guess who is here? This is Luffy. So I caught Luffy yesterday sitting on a chair in the kitchen and yawning. So I took his picture while he was yawning. So that is pretty cool. All right, so now this is our Discord server. It is continuing. So thanks to Sassy Bean, and thanks to Papa Bean, and thanks to Bonk Bonk, so and all others who are participating. Here is the links that are going to be used in this discussion today. This is drbeen.com. This is the preprint of the study that we are going to talk about, the spike proteins of SARS-CoV-2 B.1.617 and 618, and then they do the discussion. I also wanted to go over this lineage.org site. We have gone there a few times. Nextstrain.org is also interesting. Outbreak.org is also interesting. They all have the various lineages that they're tracking. And there are thousands and thousands of lineages. Then this is an interesting one. So this is how media portrays what we're talking about. And so Pfizer-Moderna vaccine effective against Indian variant. That's a study that says it. Similarly here, Pfizer-Moderna vaccine effective against Indian variants study. So it looks like when the New York University may have given a press release, and this press release is as such copied with the headings as well. This is CNN. CNN had a slightly different heading. Pfizer and Moderna vaccines appear to protect against variants first seen in India. Lab experiments suggests. Then this one is Straits Time. Pfizer Moderna vaccine effective against variants first detected in India study. And this is another one. Finally, what is an antibody titer? So this is a very commonly used word, and sometimes it is used incorrectly. And I wanted to make sure that we have that idea in our mind that what is a titer. So I have that quick article over here. So if you would like to read it, all of these links are in the discussion. Now, in this experiment, they had used a virus, which is called a lentivirus. Lentivirus is a very similar virus. It's the same species as the HIV virus. So they used a lentivirus and they modified it to produce spike proteins on it. And then they used those spike protein of this virus to see what is the efficacy against of the antibodies against that. So if you wanted to read up on what are lentiviruses, that is here. This is CDC's site. So some folks say that, hey, Wikipedia may not be that great. I hope there is some level of trust for CDC. So here is a CDC site. And so trust or not, CDC has some information about variants as well. Some of that is useful. Then this is also an important one. This is the effect of 501 N501 variant and what does it do. So that is here. This link is present too. And finally, a couple of things here. US, if you see here, number of cases are going down. Can you imagine this, that we used to have at our peak 250,000 cases on a 7-day rolling average, and we are now down to 32,000 cases. So 250,000, almost 9-fold reduction, which is excellent. And then the number of deaths are reducing in the same way as well. Daily deaths, 290. Imagine this compared to 4,000 deaths or so on daily basis. So beautiful recovery on the US side. Of course, my family and I, we kept going out on these Eid days. We had 3 days of lots of fun time. And the society is humming and people are just, they're eating and they're moving about and they're mixing and they're meeting. So I love that. The society is opening. There's a lot of criticism for CDC to jumping the gun for masks. So no wonder. Actually, CDC does not surprise me. They just keep doing these things based on their behavior about masks and about the virus transmission. If you look at their information and chaotic behavior, you can then see that if they say, we think only this drug is useful, you should see that in the same lens of their chaos as we can see with the masks and with the transmission. Here is India. So India, if you see here, they are also now trending downwards. So that is also a very good news. So with these, I'm closing these as well because they just keep some stress on the computer and it causes the computer to be slow. All right, now let's start a discussion. This initial part of the discussion, I'm going to fly over that a little faster because there is another video that I've done that says double mutant in India, and that is the same part here. This is just a quick summary of it. So 617, it actually has 13 mutations. There are a couple of mutations on the spike protein that are concerning. That is why it is called double mutation. Actually, there are 13 mutations. The 2 mutations that are concerning is one found in the UK and the other one found in South Africa. Actually, now Indian variants also have mutations that are found in California and New York variants as well, and South African variants as well. So here we have gone through this before as well. These are all the mutations that are on the 617. However, 484K and L452R, which is the California mutation, these were more concerning. And from a properties standpoint, we saw that L42R was going to reduce the efficacy of the vaccine, and that is what we're going to see today. And then 484 and 484K were going to reduce the efficacy of the vaccine, plus increase the transmissibility as well. So we'll look into that today as well. So this is all previous discussion. This is the New York study that we are seeing. So I'm going to summarize the study itself. So if you just wanted to hear the summary, then you will be happy with that and you can move on. But if you wanted to then see how did this study and what kind of percentage differences they have seen, then we'll continue to talk about that. So here is a summary. Vaccine efficacy was modestly reduced. Now, please remember this, that when we say five two-fold reduction in titer. What does that mean? So first of all, let's see what do we mean by titer. Imagine somebody has gotten a vaccine. The result of the vaccine is that they have produced some antibody quantity in their blood. And we take a small sample of the blood, let's say 10 milliliter or 5 milliliter, some measured quantity that we do the same for everyone. Now, of course, that measured quantity has antibodies in it. Now, what we do is we take a part of that blood and we react that with the antigen, for example, spike protein. Of course, there are antibodies in the blood of that person, and they would react with the virus spike protein and they will bind. And we'll say, okay, there are enough antibodies in this blood sample, small, tiny part of the blood, that it could neutralize the spike proteins that we offered. Then what we do is we take the remaining blood and we dilute it. And then once again, we take a small sample. This is like if you're cooking something, you add water to it, then you take a spoon and taste it, then you add more water to it, and then you take a spoon out of that and taste it, and so on. You keep doing it till you cannot taste anything anymore. So now what they do is they take the remaining blood, they add some diluting material to it, then they take a small piece again, measured piece, defined piece, and they once again react that with the antibodies. And if that piece also reacts, they would say, okay, we have titration of one level and the antibodies are still working. Then they dilute it again with a measured, decided quantity. So let's say put one more glass of water in the cooking pot. So I'm taking two similes. One is the actual example, one is the actual process, and I'm taking the simile of the cooking. So I hope you can keep both of those thoughts in your mind right now. So then they take another spoon from it, they take another measured quantity, and then they again have it attack the antigen. And if it still works, they say, all right, now we have a titration of 3. Then they dilute it again, and then they keep doing this process. Then they reach a point where the blood has been diluted so much that antibodies are now so little in that small spoon that when they react them with the antigen, antibodies are so less that they do not even work with the antigen anymore, and the antigen is still there. And we say, okay, we had to dilute it 500 times to reach a point where it would stop reacting. So titration level 500, for example. So if it was the cooking example, we'll say, well, we added 1 glass of water 500 times to 1 kilogram of the food, and then we reached a point where we could not taste the salt anymore. That is what titration means. So now when we say that titration reduced by 2 times, so instead of, let's say, 500, we reduced it to, let's say, 480 or 400 or 250. The actual question that should be in our mind is, what is the level that is protective more than, hey, what would 2x reduction mean? So I'm going to spill that beans right now, that when you would see here, for example, when you see here the 2 to 4 fold, the x over here is the fold, 2 to 4 fold reduced titer. That simply means that when they were diluting it, they diluted 2 to 4 times lesser, and the antibody response went away. So instead of diluting it 500 times, now they were diluting it 250 times, and 251st one did not have the response. Now the question is, do the original sample has enough response? And the answer is yes. So the day we should be concerned is when the researchers come back and say, we titrated the antigens and antibodies, and the antibodies could not work with the antigen even at the quantity that the patient had originally produced. So no titration, and still there is no binding, and that means we have an escape. Here, when you see 2.4, this does not tell the story. What tells the story is, was there still enough protective levels of the antibodies? And for all of these, you would see that they would say, yes, we have the enough protective levels. So that means, I'm going to give you one more example. This is really important. That is why I'm spending some time on it. So please don't be annoyed. I'll give you one more example. Imagine that there are 10 horses needed to pull some weight, and we have applied 1,000 horses. So now if there are 1,000 horses versus there are 500 horses, it doesn't matter, because essentially we only need 10 horses to move the weight. So 500 or 250 or 100 or 80 or 50 will not matter. We still just need 10 horses. So in us, the amount of antibodies that vaccine is generating or a previous infection is generating, that amount that can still bind with the virus and can affect it, is still large enough to take care of the virus. And then there is some amount of antibodies that are gone, that are not working anymore. So that is the reduction in efficacy. So the question always should be in our mind, that is that reduction in efficacy mean we cannot handle the virus anymore? And the answer is, we will be fine. We can actually handle our immune system from this study. Our immune system can handle Indian variants better than South African variants or even California variants. So we should not be that. The worry, if you want to be worried, if you said, you know what, Dr. Mubin, I want to be worried, tell me something that would make me worry. Then if we keep accumulating variations on top of these variations, if we keep bringing it to a point that it cannot be bound by the antibodies that we have, then we have a SARS-CoV-3, then we have a new pandemic, then we have an escape. So please keep this in mind. And I want to show you this little sentence here as well. The modest neutralization resistance of the variant spike protein to vaccine-elicited antibodies suggests that current vaccine will remain protective against the B.1.617 and 618 variants. And not only just the vaccine, the plasma and others as well. So I want to read one more sentence here, and that is, look at this. Even with a 3 to 4-fold decrease in neutralization titer of vaccine-elicited antibodies, average titers were around 1 to 500. So they diluted that 500 times, and then the efficacy stopped. So maybe they diluted it, instead of 1,000 times, by 500 times the efficacy stopped. Or instead of 3,000 time dilution, by 500 times dilution, the efficacy stopped. So instead of 1,000 horses, it stopped at 300 horses, but still it stopped. So even with the 3 to 4-fold decrease in utilization titer of vaccine-elicited antibodies, average titers were around 1 to 500, a titer well above that found in the sera of individuals who have recovered from infection with early unmutated viruses. So even better titers than the people who had the infection and recovered. Thus, so this little purple thing, thus there is a good reason to believe that vaccinated individuals will remain protected against the B.1.617 and B.1.618. I would have added one more sentence over here, and that is about the infected individuals who have recovered from this test. They also had protective effect. So this is the summary of it. Now let's look at the discussion here. So once again, vaccine efficacy modestly reduced, 2 to 4 times reduced titer. But now you know that just 2 to 4 times doesn't mean anything until you say, was the protective titer still present? And that was yes. So protective, yes. Convalescent plasma efficacy. So if you take the convalescent plasma from an already infected person and give it to this person who has 617 or 618 variant, 2 to 5 times reduced titer. So instead of 5,000 horses, after 500 horses, the weight was still working. And so there is reduction, but it was still protective. Then Regeneron antibodies. So you know that Regeneron is a polyclonal antibodies. These are actually two antibodies. And Regeneron, I believe Regeneron is such a magic thing. Trump had received it and he came out very successfully with that. Giuliani had received it, and I believe so many. And please remember, Regeneron is available in US on compassionate basis. It is authorized. You just have to ask for it, which is the weirdest thing. I think that they should just make it part of it in the viral phase to say everybody gets it, but you have to ask for it. So please remember that. So now Regeneron antibodies, out of two antibodies, one antibody could not work correctly. So that is the Regeneron. It was still protective. Reduction in titer was 4.7x. Vaccine-induced titers were 1 to 500. I just read it for you. That the titer against this variant was still 1 to 500. So that means you take the blood, which has the antibodies produced by the vaccine, and then you keep diluting it. And after 500 times dilution, then it would stop working. And maybe in somebody else with the wild type infection, let's say Wuhan version, you may have to keep diluting it till 5,000 times, and then it would stop working, and here 500 times. But the point is, it continued to take care of the virus. Now, transmissibility of the virus. With these changes in the 617 and 618, the transmission of the virus has increased by 3 times to 3.5 times. This is actually lesser than South Africa, which was about 4 times. The reason I keep comparing this is that those folks who continue to say that somehow the 617 and 618 variant is now a new pandemic and we are dead in the water, that's not the case. It has transmission that is lesser than South African transmission. It has infectivity that is similar to California's infectivity. Have you ever heard of the big hoopla about the California variant? I live here. I have never seen the news cycles about that. So I think there is some sensationalization as well that is going on. So transmissibility is 2 to 3.5 times more. Resistance extent was smaller to the earlier version. So if you see here, B.1.351, South African, and 526, New York, their reduction in resistance, or their increase in resistance, sorry, I said reduction, to the vaccine is very similar to 617 and 618. What I'm trying to say here, which seemed a little convoluted, 617 and 618 has similar reduction in efficacy of the vaccines as South Africa or New York. And we know that even with South Africa, I know that there are studies, some studies say, well, the efficacy has reduced to 8%. And then there are studies that say that efficacy has stayed above 74%. So now we have to see which one is correct. But we have South African variant in Israel as well. And Israel, almost all of it is vaccinated and they're fine. UK is fine. So I don't think that there is any vaccine so far that has ended up being useless against any variant so far. Okay, so now let's now see what are the mutations that we are seeing. So here is a spike protein. We have S1 unit and we have S2 unit. We know that with the S2 unit, we have fusion protein. With the S1 unit, we have the receptor binding domain that binds with the ACE2 enzyme on our cells. So we know this much. The 617 and 618, the changes in them that we are seeing, 617 is more prevalent, more seen as well. It has L452R change, which is unique to it. And again, I hope that you remember this. 452 is the amino acid number. Imagine if you have a long rope or you have a long wall made up of bricks, one brick each, and they're just laid out next to each other. So the 452nd brick is changed from amino acid type L to amino acid type R. So then there is another change at the number 484 amino acid, and there is an E amino acid that is changed to Q. 614, 681. So these are the four important changes. We have discussed it in the past that there are 13 changes, but these are important changes. 618, another variant in India, has deletion of two amino acids and has 484, which is very similar to 484 over here. The only difference is here the E became K and over here E became Q. And then 614, which is the very similar thing here as well. So this is similar, this is similar, this is unique, this is unique, this is unique to these two variants. Now, with these changes, what is the effect? The effect is, so if you see here on this table, this is UK or Kent variant or 117. This is, I think I wrote this wrong, South Africa 135, Brazil P1, India 617 and 618. So 501Y change. So amino acid number N change, or amino acid N changing with Y at the number 501. This change is present in UK's variant, South African variant, Brazil variant, and it increases the affinity of receptor binding domain to the receptor protein enzyme ACE2. 484K, it is present in South African, Brazilian, and India origin variants. And check this out, in the India variant, E484 increases the resistance by 2.3 to 2.5 fold. I should not call it increases the resistance. I should say titers are increased by 2.3 to 2.5 fold. South African is 3 times. And titer increasing, I should also say titer reducing. So just imagine efficacy is reducing. So South African efficacy, that variant, had more efficacy reduction compared to the 617 and 618. So should we be that worried? Yes, there is a variant, but it is not a faster variant, or it is not a more dramatically lethal variant, as some folks continue to say. And it is also not an example of a variant because vaccine failed. I have been saying it all along. This variant had emerged in India before any significant vaccination was done. E484Q, L452R, they are present in 617. They're unique to them, and they do what? They cause immune evasion, or they cause resistance in receptor binding domain. We know that receptor binding domain is important because that is what is used by the virus to bind with ACE2. So it improves the affinity of the receptor binding domain and increases the resistance of it from the antibodies, either from the infection or from the vaccine. Then 145 to 146 deletion. These are 2 amino acids that are deleted in 618, not in 617. And that interesting thing is this: it is an N-terminal antibody binding site. What does that mean? So imagine that we are making a protein. So spike protein is a protein, correct? So when we are making proteins, we are laying bricks, which are amino acids, and we are kind of making a long rope. One side of this is called an N-terminal, and the other is the carbon terminal or C-terminal. So this N-terminal is what they're talking about, that there is a mutation in this terminal, and they say that we know this terminal is something that is bound by an antibody. And so any change on the N-terminal would make some antibodies fail. And we also know this, that not all antibodies will fail. There are hundreds of antibodies that would still continue to work. So that was the 145 to 146. Then P681R change. Amino acid number 681, 617. This is a change between S1 and S2 parts of the spike protein. So if I can see a spike protein here. So let's say this is the spike protein. Remember that it has an S1 part and S2 part, and they need to be primed and separated from each other, one to fuse with the cell membrane, the other one to bind with the ACE2, and TMPRSS2 proteins do that. So that part where the separation occurs, this is a mutation in there, and they think it may help getting primed easily. It may actually be something that would resist priming. So they don't know, and in the paper they say we exactly do not know which mutation exactly does what. We know that the infectivity is increased. We know that shedding is increased. Of course, when the cells are infected more, then the virus is entering the cells and replicating and coming out and entering the next cell fast enough to create a greater viral load soon enough, and that would cause increased shedding as well. So shedding is increased by 2 times, and the infectivity is increased by 3 to 3.5, 2 to 3.5 times. So maybe this change, 61681 change at the S1 and S2 junction, is interesting from proteolysis or from breakdown of the or the priming of the S1 and S2. Okay, so now this was my little depiction. It seems busy, but it is actually very simple. Here is the spike protein. So spike protein is now ducking and covering, and it wants to avoid the antibodies that we are producing. And here is the B cell making antibodies and firing it at the spike protein. So some antibodies are not going to work because of the mutations. Others are still working. So the ones that are not working are in red over here. They're just dying there. And the others are still attacking the spike protein. Then here, if you see, this is another B cell that is attacking. Here is the convalescent plasma, and that is attacking as well. And here is Regeneron's antibody, and this poor antibody is dead. They have 2 antibodies, and one of them is dead for 617 and 618. So if you ask me that out of this whole thing, do I have any concern? My only concern is that I really love Regeneron because of its polyclonal behavior. And if one of the antibodies is not functioning, I would like them to actually change that antibody to another type, because now for Indian variant, they would be essentially as good as a monoclonal antibody in the light of this study. So then continuing, how did they do this? What was the experiment that they did? What they did was, if you see here, this happy little virus is a lentivirus. Lentiviruses are a species of virus which HIV belongs to as well. These are enveloped retroviruses. So they took a lentivirus, they added the spike protein, the mutated spike protein from the Indian variant. They added the genetic material for that in this virus. Then, as we have done this discussion many times, that how do you grow viruses? You put them in cells, you infect them in the other cells, which could be fetal cells, which could be moth cells, which could be some other cells, and then you let them grow in the cells. That is how you increase the viruses. So now they have a mutated virus, lentivirus. They put that with the spike protein, new spike protein genetic material. Then they grow these viruses in the cell. Then when the viruses come out of the cell, now they have the spike proteins on them. So first they confirm that do they have the spike protein? So how do they see that? In the supernatant or the fluids around the cells would contain spike proteins. Secondly, they would see that the viruses have spike proteins. And once they confirmed that the viruses developed these spike proteins, mutated spike proteins, then they reacted those viruses with convalescent plasma, with vaccine-generated antibody rich blood, or with Regeneron. And they saw the results which we just looked at. So here is some more data from those results. Infectivity, virus's capability to infect the next cell. L452, 484, 681, this is mostly the 617, has 2 times increased infectivity. This is actually lesser than the South African variant's infectivity. L452R has 3.5 times more infectivity. So that alone, one mutation can actually bring it up to 3.5x. If it is 3.5x, then it is slightly more than the South African variant. L452R and 484Q together, 3 times infectivity. So various mutations, as you can see, some mutations together are reducing the infectivity and some are increasing the infectivity. All mutations are not always useful. There are many mutations that are not useful. Normally, those viruses are not selected or they're not used any further because they're disabled viruses. And those that are fit, that are better, they would continue to live on and continue to spread. Again, I would maintain this, that as they would spread more, they will become more adapted to our system as well, and they would cause less lethal behavior. So this is the infectivity. I was going to say with the less lethal behavior that I understand that as they spread more and the window of infection is reduced, our chances to quickly get ahead of that and treat the patient may not be correct. So more people would die as a result because of spread. But the lethality of the virus itself is actually reduced. Neutralization of the convalescent plasma. So once again, if you see here, this is 617, 617, this is 618. So 2.3 time to 2.5 time or fold reduced titration. Again, was it effective still? Yes. Was it less effective? Yes. Was that reduction in efficacy too much? No, it was a modest reduction. Neutralization by the mRNA vaccines. So they say in their study that, hey, we have not done the study on non-mRNA vaccines. So here, the analysis in this study were restricted to the messenger RNA-based vaccines, but there is no reason to believe that vector-based vaccines, adenovirus-based vaccines, such as that of Johnson & Johnson that express stabilized native full-length spike protein, would be different with regarding to antibody neutralization of virus variant. So they're saying others should behave the same way. So back here to the discussion. So neutralization 617 had 4-fold increased resistance for titration. 618 had 2.7 times more titration need, so resistance. So I shouldn't say more, I keep saying it more. Reduced titration. So instead of diluting it 500 or 5,000 times, now we could only dilute up to 1,000 times, for example, and then 1,001st will not work. So it is actually the titration number reduced, which means efficacy reduced, which means resistance increased, but still not enough to escape. So here still 1 to 500 titer. That means for up to 500 dilutions and it still works. The antibodies produced still work. Then L452R mutation. This mutation is actually California mutation, which is also observed in the India variants, so 617 and 618. That is a primary culprit for increased shedding, increased resistance and shedding. So how much increased shedding? According to the paper, 2 times more shedding. That means the load increased 2 times more than the wild type. So they're expecting that if it is increasing by 2 times, then it is shedding twice as much as well. 4 to 6.7 time reduction in antibody titers for convalescent plasma. This is the 452R change. And 2 times reduction in antibody titers for vaccines. So what is the summary once again? The summary is they have observed that the 617 and 618, the variants originated in India, have caused they have increased resistance, and the neutralizing antibodies from convalescent plasma or the vaccine have reduced efficacy. But that efficacy is not reduced enough to say that the virus cannot be managed by these vaccines. So even if there are no vaccine modifications to target to the variant, we are still fine. That also means, I know that some folks would be saying that, hey, somehow Mubin is just trying to talk about the vaccines and it is selling vaccines. Some people now actually say, I get some money from Big Pharma to promote vaccines. So uh, there is no money, no big pharma either. But anyways, uh, normal infection. So those who are saying, you know what, I just want to have the normal infection, dangerous thing to do. It has a really serious outcome of either a long haul or death, if at all, in somebody ends up in that direction. Majority would survive, correct? So even somebody who has recovered from a normal infection or wild type infection, their efficacy is similarly important. So it is not just the vaccine. And an extra bonus is they tested Regeneron as well. So this is a discussion. I'm going to show up again in 5 minutes for our chit chat. So closing comments once again. So please like, subscribe, and share. If you don't want to subscribe or share, then just like it. Number one. Number two, if you would like to support this work, you don't want to use PayPal, there is a link to use for buying me a coffee, or you can become a patron. If you're okay using PayPal, there is a link as well for PayPal too. So with that, thank you very much, and I would see you in 5 minutes.